Hi, I'm Carles, a postdoctoral researcher in the lab of Patrik Verstreken at the VIB-KU Leuven Center for Brain and Disease Research, and I would like to tell you a bit more about our Brain-on-Chip project. At our center, our goal is to find cures for neurodegenerative diseases by studying its underlying biological mechanisms. Parkinson's is one of such neurodegenerative diseases that affect over 6 million people worldwide. The disease mostly causes motor symptoms such as tremor and slow movement. Currently available treatments only manage symptoms and do not actually cure the disease itself. There are several barriers to effectively studying what goes on in the brain of Parkinson's patients. Unlike other organs, living brain tissue is very difficult to access. The more detailed neurobiological analysis on the brain can only be done on postmortem brain tissue. This is limiting in that it only provides a snapshot of the disease. We can only observe it at the very end, once the damage is already done to the brain. And we cannot see the processes that trigger and drive the disease from the beginning. We are lacking an early diagnostic tool that could enable us to identify people who are in the earliest stages of developing brain disease, well before any major damage has taken place. And that's where Brain-on-Chip comes in. We want to be able to define Parkinson's disease at the molecular level, rather than simply by clinical symptoms of the disease, which are usually observed when it's too late to intervene. To this end, we are creating 2D and 3D brain-on-chip models to recapitulate the neurobiological basis of the disease in the lab. And how do we do this? We use skin cells from Parkinson's patients from cell banks at Stanford University. We reprogram these cells using transcription factors into the specific brain cells that are affected by Parkinson's. We then use these specific brain cells to recreate the neuronal networks that resemble the areas most affected in the brains of Parkinson's patients. Then we grow these networks on a microchip which contains multi-electrode arrays, which we can use to manipulate and measure electrical activity of cells in the circuit. This will help us understand more about what they do. This method allows us to go back in time to the earliest stages of Parkinson's. We can use this tool to compare cells from patients versus cells from healthy controls in order to better understand more about what goes wrong in the brain early on the disease. We can also use this method to divide patients into subgroups based on their neurobiological signature, with the ultimate goal of developing personalized therapies that target their unique molecular and genetic makeup.